focus. Hello, physiology. It is a real pleasure to have Dr. Xiaochen for physiology seminar today. Since everybody probably knew him very well, but I prepare for introduction, so I go through the introduction. Dr. Xiaochen studied precision instrument and mechanology in Tsinghua University in China and continued his PhD in biophysics under supervision from Professor Zhang Fengxi in the same university. During the time, his study was on biomembrane and membrane biotechnology. Following his PhD, Dr. Bai moved to MRC UK 2012 for further training on protein function and structures using cryo-EM. During the short 3 years in MRC, published the most challenging cryo-EM structures, including ribosome complex, human gamma secretase, iodine receptor, and also developed a method for advancing cryo-EM technology. It is a great luck for us. 2017, he set up his research lab in biophysics department at UT Southwestern and helped to build a large research community using cryo-EM application in campus. His contribution is proved by publication with many collaboration in campus. In parallel, his own lab continued to produce many articles on very challenging protein structures to understand their functions. He told us his current research focuses on structure and functional determination of receptor tyrosine kinase, signaling of sting and scab, and ion channels and transporters. But above all, for my favorite part of him is when I meet him in elevator, I always greet with a very kind smile. And that is my favorite part of him. Today, we can hear one of his recent work titled Structure Basis of the Activation of Insulin Receptor. Please welcome Dr. Bai.
Yeah, thanks, Minwei, for really kind introduction. And also want to thank Lilia and Minwei for very kind invitation. Yeah, it's really great for me to have the opportunity in physiology to share some of our recent progress in understand how the insulin is activated insulin receptor. First, let me share my slides. Okay, yeah, before I start talking about the insulin receptor, I want to give you a brief background introduction about what is the major project in my lab. So our lab is, as Minwei mentioned, we try to understand and how the ligand can activate a large family of receptors. It's called receptor tyrosine kinase. So that is from here. This is a very simplified model about how the receptor is activated. For most of RTKs, the ligand is mostly a dimer. The ligand forms a stable dimer, and the dimeric ligand can recruit two receptors and to induce called a receptor dimerization. So in the dimerized receptor, the two intracellular kinase domain can meet each other and therefore undergo this so-called autophosphorylation. So in the C-terminal of each receptor, there are normally a group of tyrosine residue can be phosphorylated. So the phosphorylated tyrosine residue can subsequently recruit many downstream effector protein and thereby activate the downstream signaling cascade. But I want to say this is a very, very simplified model for the activation of RTK. And there's actually a lot of RTKs in this family. And you can see from this figure, although RTK share very similar structure features, so for most RTK, they only has a one transmembrane helix. That's very different to other receptors like GPCR. GPCR has 7 TM, but in those receptors, it only has 1 TM. And also, old RTKs has a similar intracellular kinase domain. But you can see the structure of outside the cell. They are quite different, right? They are all the structure in the actual cellular region, and they are very diversified. So that already suggests the activation mechanism among different RTK could be quite different. And also there's some quite special RTKs. The ligand is not enough to activate the RTK. So for example, this is through the collaboration with Xiaochen Zhang from pharmacology. We determine the structure of a ligand and the receptor. This is called RAD receptor. So we determine the structure of ligand, co-receptor, and RAD complex. So in this case, the ligand is not enough to activate receptor. It's also required co-receptor. So the three proteins need to form a ternary complex for the activation. And this is also very recently also working with Xue Wu's lab. So we actually determined another RTK structure called CMAT. So it's different to most of RTKs. The ligand for CMAT is called HGF. And HGF is a very large molecule and always stay in the monomeric state. In them, it cannot be from a dimer. So from this structure, you can see the one HGF is sufficient to recruit 2 CMAT for that dimerization. So the one HGF can use 2 different interfaces recruit 2 CMAT. And this is also quite unique mechanism in activation of RTKs. But today, I want to focus on, I think it's one of the most famous RTK is the insulin receptor. So in this insulin receptor, actually is comprised of small family. In this insulin receptor subfamily, it totally has 3 structure-related receptors. The first one is the insulin receptor. It can be activated by insulin. And the second one is also quite famous. It's called type 1 insulin-like growth factor receptor for IGF-1R for short. So IGF-1 receptor can be activated by IGF-1. And the third one is relatively less well studied. It's called insulin receptor-related receptor. I think the name is quite confused. This name is very similar to insulin receptor, but they are different receptors. So among those 3 related receptors, they share about 50% sequence identity. Actually, you can see the 3 receptors are very similar. But I want to maybe in my talk, I will focus on the insulin receptor, but also I will briefly touch at the IGF-1 receptor. And if I have time, I will briefly also mention the activation mechanism of IRR. Yeah, actually, also the activation mechanism are very, very different among those 3 related receptors. Okay, so actually last year, it's actually the discovery of the insulin for 100 years. Yeah, it's quite amazing, right? The insulin has been identified for 100 years. This is a picture of Charlie Bass and Frederick Betting. They actually used the diabetic dog to really understand the action of insulin. So yeah, but think about it, it's also quite interesting. After 100 years, people still don't really fully understand and how the insulin is activating the receptor. And the reason behind actually is the way insulin activates receptor is quite complicated. Firstly, actually insulin, as I mentioned, can be activated by the insulin receptor, can be activated by insulin, and those activation will leading to the AKT pathway, and the cell use the insulin-activated insulin receptor to control the glucose uptake. But in the same time, insulin receptor is actually can also activated by the IGF-1, IGF-2. So IGF-1 and IGF-2 is also a very small peptide. Structurally, it's very similar to insulin. But when the insulin receptor is activated by IGF-1 or IGF-2, the signaling consequence is very different. So actually, the IGF-1 activated insulin receptor can activate the ERK pathway and ultimately will control the cell growth. So, but there's another receptor I mentioned is IGF-1 receptor. That activation is relatively simple because IGF-1 receptor can be only activated by the IGF-1 and IGF-2, and in this way, it can control the cell growth. You can see the insulin receptor actually is a multifunction receptor and can respond to different ligand stimulation. So yeah, it's very intriguing question how the different ligand actually can promote a different signaling response through the same receptor, right? This people call this BIOS agonism in the field, and how I think we try to understand from the structure biology how this happens. Another complication in the insulin receptor is the insulin actually activate when the insulin activates receptor, people have shown that the binding between the insulin and receptor and the insulin seems to involve a lot of complication and very complex. And for example, this is the people has done this binding competition binding experiment almost 50 years ago. So for this called a scatter plot, that is a competition binding experiment. They're showing when they use insulin doing the binding experiment with a receptor, the binding curve is not a linear shape, right? This is called curved linear shape. That actually indicate there must be a multiple binding site on the receptor can bind the insulin, and those binding sites may have a different affinity, and most also may involve coercitivity, right? That is make the binding of insulin to receptor seem to really complicate. And because insulin is a relatively small molecule, and in total is about 50 residue. So people has doing this called alanine scanning metagenesis to identify the residue of insulin that is important to bind to site 1 or site 2. So it's really very clear. This is a picture showing, yeah, the one side of the insulin are important to the binding to site 1 of the receptor, but the opposite side, and that's showing in purple here, is important to the binding to the site 2, right? But this is only on the insulin side because receptor is very, very large molecule. You cannot do such an alanine metagenesis on the receptor side. So now actually where the site 1 and the site 2 on the receptor is not very clear. So this is a domain topology of the insulin receptor. So different to all other RTKs. So insulin receptor form a stable dimer, and those dimer is linked by multiple disulfide bonds. So they never falling apart. So even without ligand bond, insulin receptor is still in a dimer form. Yeah. So this is a very, very important feature for the insulin receptor. So in each proteomer, there's a multiple domains. There's L1 domain, CRD domain, L2, and the 3 consecutive fibronectin 1 domains. But you don't need to remember the name of these domains, but I hope you can remember one important feature of the insulin receptor is during the maturation of insulin receptor, each single chain of insulin receptor will be cleaved in the trans-Golgi by a protease called furin into 2 chains, we call alpha chain and the beta chain. So in the C terminal of the alpha chain, there's a small motif called the alpha CT motif. So alpha CT motif is, you can see sequence-wise, it's very, very far away from the L1 domain, but structurally, L1 domain and alpha CT motif will make a very strong interaction. They always stay together. So please remember this feature so that the alpha CT motif is always bound to the L1 domain, although they are separate on the sequence. And so structurally, alpha CT motif is a part of the alpha CT motif is folded as a small alpha helix. So and another important feature is the 2 alpha CT motif is also linked by 3 pairs of disulfide bond, right? And actually, I will show you in the later of my talk So the disulfide link alpha-CT motifs actually are very important for the regulation of the insulin receptor. So, and the crystal structure of the insulin receptor in the apo state has been determined by X-ray crystallography a long time ago. This is the work from Australian group. Actually, the overall structure is an inverted wave shape, and this I colored this two pertamer in two different colors. As you can see, the two insulin receptor pertamer actually tightly associated with each other and form a very stable dimer. And the interaction is mostly between the L1 domain of one pertamer and the 5-nectin-2 domain of the adjacent pertamer. But from this structure, you can tell because the two legs of the insulin receptor are separated by long distance. Therefore, the two intracellular kinase domain cannot be adjacent to each other because of the long separation of the two legs. So this suggests, so this is apo structure of insulin receptor represent the receptor in the autoinhibit state. So this is the inactive structure, right? Because of the separation of the two legs. Okay, so and this is another. The previous apo state is published in 2006. So after 7 years, this is by the same group. They also saw the structure of the insulin bound with a truncated insulin receptor domains that is only contains the L1 domain and also the alpha-CT motif. Yeah, remember I told you alpha-CT is always stay together with L1 domain. So actually the L1 domain plus alpha-CT motif serve as an insulin binding site 1. So this is clear how the insulin is bound to the site 1 of the receptor. And the binding mode has been clearly reviewed by the X-ray crystallography. And showing the insulin they actually make a simultaneous contacts with both alpha-CT motif and also L1 domain. This is very, very clear. And very important in the recent, because of the recent resolution revolution in the QREM, people started to use QREM to understand how the insulin is activated with receptor. And this is the structure is published a couple of years ago by company Merck. So they actually using the actual truncated extracellular region of insulin receptor, and they actually saturated with insulin. But you can see the structure is quite different to the apo state. The apo state is inverted wave shape, but this structure is more like a T-shaped receptor. And they actually also identified the binding of the insulin to the site 1 of the receptor, and the binding mode is very similar to that has been observed in the crystal structure. But again, the problem for this structure, they actually didn't see the site 2 binding. And also the overall structure is quite flexible. And this structure is unlikely to represent the fully active state of the insulin receptor. The reason actually people cannot capture the receptor in the fully active state, and it's because you should not only use the actual cellular region of the insulin receptor for the structure study. That is because people has done the binding experiment. They show that insulin bound to the full-length receptor is in a much stronger affinity as compared with binding to only the actual cellular domain. And the difference in the binding affinity is almost 1,000 times. Therefore, if you only use the truncated actual cellular domain of insulin receptor, it's unlikely you can capture the fully active state because the binding affinity is much, much weaker, right? So that is why we started working on this project. So we want to really purify and the full-length insulin receptor for the structure studies. So in this way, we may have a chance to capture the receptor in the fully functional state. And also and this is the question we want to answer before actually we started working on this project. So firstly, where is the site 2 on the receptor? But now only the site 1 has been identified by the X-ray crystallography. And why the activation of the receptor require multiple binding sites? right? What is the exact activation mechanism? And I mentioned this in the very beginning. So insulin receptor can be activated by both insulin and IGF-1 and IGF-2. And how the different ligand, IGF-1, IGF-2 insulin, can lead to different signaling outcome through the same insulin receptor. Right? And lastly, we also want to understand IGF-1R, which is very related to insulin receptor, is their activation of mechanism of IGF-1R similar or different to insulin receptor? Right. Hopefully, I can give a brief answer after my talk. And before I started, I want to thank the people working in the project. So Emiko Ishikawa, Jie Li, and Li Wei Wang is three people from my lab working on this insulin receptor project. And I want to give my very special thanks to Yunhee Cho from Yunhee is a postdoc from Hongtao Yu's lab. And actually, we started working on this project when she was a postdoc here. And Yunhee is a really excellent and cell biologist. So she actually helped us to perform all the functional experiments. And now Yunhee is the assistant professor in Columbia University. And also I want to thank Xuewu Zhang for really a lot of this very useful discussion. And that is really, I learned a lot of knowledge from Xuewu on the RT case. Okay, so as I mentioned, we want to start the receptor using the full-length receptor, but actually it's quite challenging to purify the full-length receptor, mostly because the yield for the full-length receptor is always relatively low to have enough protein for the structure study. So one trick, so we have to mutate the residue on the receptor, which is important for the endocytosis, because once the receptor is activated, the receptor will quickly get internalized through the endocytosis. But if we mutate the residue that actually controls the endocytosis, we can increase the yield for almost 1,000 times. That really helps us to get enough protein for the structure and the functional study. Yeah, another trick actually is a postdoc from Xuewu's lab. He developed this new affinity purification system called using the strong binding between the TSE3 and TSI3. This is a toxin and antitoxin protein. Because the binding affinity between the two proteins are really high, it's more picomolar affinity. So actually we can really fish a lot of protein from the cell, even the expression level is still not so high. So with the help of these two tricks, so we can get enough protein for the structural EM study, and you can see that we can get a relatively homogeneous sample, and we image the sample under our microscope. So after 2D average, we can already see the 2D average looks pretty good, and we can see the receptor in different orientation. And here you can see there's a micelle, actually that is where the transmembrane domain of the receptor. But unfortunately, from the 2D average, we cannot see any density corresponding to the kinase domain of the receptor, suggesting the kinase domain is highly flexible and can average out during the single particle image processing. And but still, yeah, we can determine the full-length insulin receptor insulin complex structure at quite a high resolution, at 3.2 angstrom resolution. And as I mentioned, the TM and the kinase domain was resolved in a very poor quality. But after using this focus classification approach, we can kind of see some density for the transmembrane helix. So actually, the two single pass TM actually dimerize also in the detergent micelle, and we can clearly see that the TM density as kind of as a sausage-like density. But again, this is not in a good resolution for the atomic model building. So yeah, but the most interesting part for the structure, because we use a full-length receptor, we really capture the receptor in the very fully ligand state. So, and the overall structure is a very homogeneous T-shaped receptor, and the overall structure has a twofold symmetry. So from the insulin receptor dimer, so we can totally identify four different insulin in the receptor. So yeah, and the two insulin are bound. They show in yellow here, and are bound in the top part of the receptor. That is at canonical, we call this canonical insulin set 1. And another two insulin actually was firstly observed, and no one has seen that before actually. And the binding site of another two insulin is kind of in the middle part of the receptor, that is on the outer surface of the fibronectin 1 domains. So, and we think this is actually the two new insulin binding site is site 2, is for the on the receptor for the insulin receptor binding to the insulin is site 2. So quick summarize. So we can show four insulin binding here, and the four insulin, two are bound at clinical site 1 and two are bound normal site, and these four insulin are related by the 2,4 symmetry. And people know actually the site 1 binding is very, very critical for the insulin activation. But from our structure, we can understand also why the insulin binding to site 1 is important for the activation. So from our structure, actually, we can see that actually insulin bound at site 1 in the active state simultaneously contact two pertamers. So in this way, the binding of insulin at site 1 can stabilize in the T-shaped active state of the receptor. So we call this because it simultaneously contact pertamer 1 and the pertamer 2, and we call this a contact site at pertamer 1, we call site 1A, and contact site at pertamer 2, we call site 1B. So if we mutate the residue from both site 1A and site 1B, you can see this is a mutation on the receptor side, and the single mutation can almost completely abolish insulin-dependent receptor activation. So that suggests the insulin binding to the site 1 is very critical for the receptor activation. Okay, so, and actually, our structure analysis also suggests there's another role for the insulin, for the site 1 insulin binding. So as I mentioned, in the apo state of insulin receptor is an inverted wave shape. The apo conformation is stabilized by strong interactions between the L1 domain of one pertamer and the fibronectin 2 domain of another pertamer. So the binding of the insulin to the apo state, to the site L1 domain in the apo state actually will clash with the fibronectin 2 domain of adjacent pertamer. That actually suggests once the insulin bound to the apo state, it will disrupt the apo conformation. And this actually becomes very important driving force for the conformational change between the apo state to the active state. So because apo state is so stable, they need the insulin binding to disrupt. So that is actually the model we have for the insulin binding to the site 1. So the binding of insulin to site 1 to the apo state will destabilize the apo state, and thereby can trigger the conformational change between the two pertamers. That is a complicated motion that mostly involves the rotation between the two pertamer and also hinge motion, which is within each pertamer. So, and therefore they can trigger the conformational change and allow the insulin receptor to form this T-shaped active state. So in the T-shape, the two legs of the receptor has been placed in a very close distance. The distance between the two legs has been shortened from 120 angstrom to about 30 angstrom. That actually allowed the intracellular kinase to make interaction and to undergo this autophosphorylation. So that is, we believe, the role of site 1 binding. So the site 2 is we actually is identified for the first time, but because the binding of site 2 is only on the one surface, it doesn't involve the kind of the cross-linking between the two pertamers. So the surface is on the the outer surface of the fibronectin-1 domain. So from this structure, it's really unclear why the SET2 binding is important for the activation. But we know this is important because if we mutate the residue on the SET2 surface, we introduce about a lot of different point mutation. If we mutate those residue, all of this point mutation can lead into weaker activation of the receptor when in response to insulin. So you can see the older mutation that introduced on the SET2 surface can lead into a weaker insulin response. So, but they're different to the SET1 mutant. For SET1 mutant, the receptor is almost died. There's no activation at all. But for the SET2 mutant, there's still about 50% activity left. So the receptor is not completely died. There's still activation, but weaker. That actually suggests SET1 is critical for activation, but SET2 may play a regulation role during the receptor activation. So, but again, from this single structure, we really do not understand why the SET2 is important for the activation of the receptor. To really understand, to answer this question, actually we collaborate with Michael Starwar from Colorado. So he's the expert to make a synthetic insulin analog. So the idea is if we can mutate the residue on the insulin receptor, and we can control the binding of insulin to either the SET1 or SET2. So in principle, we can introduce mutation to the SET1 residue on the insulin, and those insulin can only bind to SET2. And in the same way, we can introduce a mutation to the SET2 residue of the insulin, and those insulin can only bind to SET1. So we call this a site selective insulin mutant. So we design a series insulin mutant that can only bind to either SET1 or SET2. So for those insulin can only bind to SET2, we call the SET1 mutant. And for those insulin which can only bind to SET1, we call this insulin SET2 mutant. Okay. So firstly, we confirm those insulin are indeed site selective. So we perform the ITC experiment, and you can see the insulin SET1 mutant, insulin SET2 mutant can still bind to the insulin SET1, but cannot bind to insulin SET2 anymore. Similarly, the insulin SET1 mutant can still bind to insulin SET2, but cannot bind to SET1 anymore. So just confirm the mutation works. Okay. So we first determine the structure of the insulin receptor with insulin SET1 mutant. Those insulin can only bind to SET2. So the structure reveals that the SET1 mutant can bind to the fibronectin-1 domain of the receptor, and the binding mode is very similar and to the insulin binding in the T-shaped active receptor. But if we compare this structure with APO structure, you can see if the insulin receptor with only the SET2 binding, the conformation of the receptor is very similar to the conformation of the APO state. So that suggests, so although insulin can bind to SET2 in the APO state, but only the SET2 binding cannot induce any conformational change of the receptor. Right? So this is also consistent with our cell-based experiment. So if you use this is a violin A3E mutant, that insulin cannot bind to the SET1. So if you use that insulin to simulate the cell, it cannot activate the receptor at all. So there's no activation. That is consistent with our structured data. Okay, but it's very different when there's insulin only bound to SET1. Right? So we can use SET1, SET2 mutant, and those insulin can only bind to SET1. So, but the structure, the overall structure is also different to the receptor with a wild type insulin receptor, wild type insulin binding. So remember in the wild type receptor, wild type insulin structure, the whole conformation is a T-shaped symmetric conformation with 4 insulin bound to both SET1 and SET2, right? But in this structure, because insulin can only bind to SET1, and actually the important information we learn from this structure is the majority of the particle actually adopt asymmetric conformation. Is you can see 80% of particle actually adopt this asymmetric conformation. Only 20% of the particle adopt this symmetric conformation, right? This suggests although insulin binding to Sat2 can trigger the conformational change from the apo state to the symmetric T shape, but much weaker capability compared with insulin wild type. Right? In the insulin wild type, all the particle go into the symmetric conformation. But if the insulin can only bind to Sat1, so only 20% particle go into symmetric state, and the majority actually is trapped in this we call this asymmetric conformations. So sorry, from this structure you can see actually both structure actually is clearly this is not the inactive state because the two legs in both asymmetric and symmetric conformation are very close with each other. That suggests there must be some partial active state. So that is also confirmed by our functional experiment. So if we use this insulin Sat2 mutant to stimulate the cells, indeed, actually the receptor can be activated to some extent, right? If you only use the Sat1 mutant, there's no activation at all. But if you use Sat2 mutant, there is some activation, but again, it's weaker than that by the wild type insulin. This is also consistent with our structure data. So if you look at the asymmetric conformation closely, this actually the structure is quite interesting. So in half of the receptor, the insulin binding at the Sat1 is very similar to the binding mode in the T-shaped symmetric conformation. But however, in another half of this asymmetric structure, the one Sat1 bound insulin also weakly touch the Sat2, right? So we call this this is a hybrid binding site. So the one insulin primarily bound at Sat1, but in the same time weakly touch the Sat2. And the way this Sat1 bound insulin touch Sat2 is different to the insulin bound at the Sat2 in the T-shaped symmetric conformation. So you can see this white insulin is, we call this clinical insulin binding at Sat2. But the yellow insulin is the insulin bound at Sat1, but also weakly contact Sat2. So the two binding mode is very different. And also apparently the binding affinity is also different because for the canonical Sat1 binding, the barrier surface is about 500 and 800 angstrom, square angstrom. But for this Sat1 bound insulin, the barrier surface is only at 360. Therefore, the Sat1 bound insulin is the binding affinity to Sat2 is much weaker than the canonical Sat2 binding. So, and actually, all the data suggests so far that if there's a binding of 4 insulin to both Sat1 and Sat2, the receptor from this asymmetric conformation. But if there is only the Sat1 binding, most particles are trapped in this asymmetric conformation. That actually suggests binding to both Sat1 and Sat2 is required to really overcome the energetic barrier associated with asymmetric conformation. And therefore, it's very important. Sat1 and Sat2 binding together is important to promote the formation of the T-shaped symmetric conformation. Okay, so to test this idea, so we do another experiment. So we mixed both insulin Sat1 mutant and Sat2 mutant together for the acro-EM analysis. So in this experiment, we incubate 1-to-1 ratio of insulin Sat1 and Sat2 mutant. And then we incubate those mutant insulin with a receptor and then subject to pro-EM analysis. So now actually, after we determine structure, there become only one conformation. It's only we only see single conformation, which is a T-shaped symmetric conformation with 4 insulin bound on both Sat1 and Sat2, right? And we compare this structure with the insulin receptor with a wild type insulin bound, and they are almost identical. That actually support our hypothesis that the insulin binding to both Sat1 and Sat2 is required for the promotion of the T-shape with symmetric conformation. So, and again, this is also consistent with our cell functional experiment. If only at the insulin SET1 or SET2 mutant, both cannot activate the receptor. But if we mix SET1 and insulin SET2 mutant together and use that mixture to simulate the cell, now actually the receptor can be activated in a similar level as that by the insulin wild type. This is right. If you mixture the SET1 to mutant, you can rescue the receptor activation. And yeah, that is just a quantified result. The receptor cannot be activated fully by both SET1 mutant or SET2 mutant alone, but it can be activated to the fully active state by the combination of SET1 and SET2 mutant together. And this is also, we also perform this called insulin tolerance test. That is, you inject insulin to the mouse. So if you only inject insulin SET1 mutant or SET2 mutant alone, the glucose level of the mouse cannot be reduced. But if you inject the SET1 insulin or SET2 insulin mutant together, the glucose level can be reduced to a similar level as compared to those mouse injected by insulin wild type. That actually suggests the insulin SET1, if there's binding to the both SET1 and SET2 together, can in synergy promote the full activation of the receptor. Okay, so because this is all the data so far, and if there's only SET1 mutant bound, SET2 mutant bound, there's all the majority of the conformation, and it's trapped actually in this asymmetric conformation. So actually, we wonder maybe at lower concentration of insulin, when the insulin cannot fully occupy by the when the insulin receptor cannot be fully occupied by insulin binding, maybe the conformation is similar to the structure of the receptor that is only occupied by the insulin receptor, insulin SET2 mutant. So because yeah, at low concentration of insulin, maybe the SET2 maybe it's not occupied, though the conformation can be similar to the receptor and insulin mutant complex, right? To test this idea, also we determine the structure between the insulin receptor and also a subsaturated insulin concentration. So we add 2 insulin receptor and we mixed 2 insulin receptor and 2 insulin in the solution, right? Because the full occupancy is need 4 insulin for each receptor dimer. So, and now we only add 2 insulin per each receptor dimer, right? And to see what happens. And this is also very, very similar to the result we got for the insulin receptor and the insulin SET2 mutant. So only the 20% of the particle going to the symmetric T-shaped conformation, and the majority of particle actually adopt this asymmetric conformations. But this is actually quite a bit surprise to us. We actually observe 2 different kind of asymmetric conformation in this data set. That is different to the structure of insulin receptor with SET2 mutant bound. And so the big difference is we call this for the conformation of 1, that structure is very, very similar to the receptor with insulin SET2 mutant bound, right? There's one insulin mainly bound at SET1, in the same time also weakly contact SET2. But we also observe another conformations. So in this asymmetric conformations, and actually this insulin actually primarily bound at SET2, but in the also same time simultaneously contact the SET1. Right? This is alternative binding. So because this is the reason this can happen is this we call this hybrid site, right? The SET1 and SET2 are really close with each other. So that allows the insulin to kind of has a 2 binding mode, right? The insulin can mainly bind at SET1, in the same time contact SET2, or can mainly bind at SET2, and in the same time weakly contact SET1. Right? This is made possible because the 2 sites are very close with each other. Right? But overall, the 2 asymmetric conformation is has very similar overall shape. Yeah, so that is a briefly summary so far. So if the insulin receptor are stimulated at the insulin site 2 mutant or at a substrate insulin concentration, most of the particles are from this asymmetric conformation, and this is a partial active state. But if insulin is stimulated by both site 1 and site 2 insulin together, or at a very high saturated insulin concentration, all the particles go to the symmetric conformation, right? And in the T shape and with full insulin bound. That actually suggests, so the asymmetric conformation cannot be formed if the insulin receptor is fully occupied by full insulin, right? And actually our structure can explain why this happens. And I show you why when the insulin are occupied by full insulin, they cannot form asymmetric conformation. So again, I go back to this asymmetric conformation where there's two alternative insulin binding in this asymmetric conformation. So the site 1 bound insulin can also weakly contact site 2, and the site 2 bound insulin can weakly contact site 1. You may ask why you can ask, maybe in principle, another insulin should be able to bound here, right? Because there is two sites here. Yeah, in principle, one insulin can squeeze in here, and the two insulin can bound site 1 and site 2 in the same time, right? Because there is two binding sites is available, right? And you may ask why this cannot happen. Yeah, now it's mostly because this. Remember, I show you this in the introduction. There's the two disulfide bound linked FCT motif in this asymmetric conformation from a very stretched conformation. Yeah, because the two FCT motif are linked by disulfide bound and so stretched, and they are linked to two site 1s. So that actually provides the conformational chain of this this blue site 1, right, to go outward. So therefore, because the disulfide bound FCT is stretched and provide conformational change of the blue site 1, it cannot provide enough space for the binding of another insulin to this hybrid site, right? Yeah. So in principle, if the FCT motif is not linked, and if the L1 domain can just easily move straight outward, and that allow the binding of another insulin to this hybrid site. But because FCT motif is so stretched and provide the conformational change of the blue site 1, so it cannot allow another insulin bound to the hybrid site. So to test this idea, we can simply remove the disulfide bond between the two FCT motifs. And now actually, sorry, I showed the idea. Yeah, when the old insulin are all occupied in this four site, because of the stretched FCT motif, the two insulin will clash in this asymmetric conformation. That is explained when the older insulin site are occupied by insulin, it cannot form this asymmetric conformation. It has to form this symmetric conformation. Right? But think about if we remove the FCT motif, the disulfide bond between the two FCT motifs, that the structure restraint will not be happen anymore. Right? That may allow the insulin receptor to form asymmetric conformation even in the fully occupied state, right? Indeed, actually we do the experiment. So if we remove the straight disulfide bond between the two FCT motifs, now we can observe most of the particles actually adopt this asymmetric conformation. And even at a very saturated insulin concentration, right? And we can see actually the two insulin are actually bound in the both site 1 and site 2 in this hybrid site, in 1/2 of the receptors. That actually suggests the FCT motif actually is important to promote conformational change from asymmetric to symmetric. Yeah. Okay, so yeah, you can see there are two insulin actually can bound in this hybrid site if we remove the disulfide bond between the two FCT motifs. So that's the summary. So I hope I can convince you. So at low concentration of insulin concentration, the receptor can adopt many different asymmetric conformation, and there's possibility they can also form the symmetric but compared to the wild type high concentration of receptor, it's only a small portion of the receptor go to the symmetric conformation if the concentration of insulin is very low. The majority of the particle, when the concentration of insulin is low, actually is adopt this asymmetric conformation. So but at high concentration of insulin or at saturated insulin concentration, all the particle need to go to the symmetric conformation. This is because the asymmetric conformation, it cannot be formed when all the sites are occupied by the insulin. That's mostly because the two insulin will be clashed in the symmetric conformations and due to the stretch conformation of the RCT motif. Okay, so that is the. So we believe the reason the nature developed such a complicated system is because that allowed the insulin receptor, because the insulin concentration vary a lot during a day, right? After eating, after taking exercise, the concentration is always changed. So having a such multi-site binding system, and as an insulin receptor can adopt different conformation with different number of insulin bound, that actually allowed the insulin receptor to respond a large concentration of a large concentration range of the insulin, and they can have different activity when they're different at different concentration of insulin. This is different to the switcher model, right? We call this tuning. You can tune the light and not really turn on and turn off the light. And that actually that is very different in the case of IGF-1 receptor. So in the IGF-1 receptor, we also saw the structure of the IGF-1 receptor with IGF-1 bound. So in this case, actually, even an IGF-1 cannot bound to the site 2 of the IGF-1 receptor. So in this case, even at a very saturated IGF-1 concentration, actually we can only see the one IGF-1 is bound on the receptor and forming this asymmetric conformation. That actually suggests there's a very strong negative cooperativity between the two site 1s in the IGF-1 receptor. That is kind of very different to the insulin receptor case. So, and the negative cooperativity of the IGF-1 receptor is actually also caused by the disulfide linked RCT motif. In this case, the RCT also form a very rigid conformation that actually kind of strongly couple the two site 1 that prevent the binding of another IGF-1 to the we call this an unligand site 1. Right? This RCT motif actually causes negative cooperativity between the site 1 in the IGF-1 receptor case. So if we remove the disulfide bond between the two RCT motif in the IGF-1 receptor, or we can make the IGF-1 RCT more flexible, and we can see more binding of the ligand to the IGF-1 receptor. And structurally, we can also become the symmetric conformation because the negative cooperativity is abolished by the mutations. So that actually quite interesting. So in the insulin receptor case, the FCT play a role in promote the T-shaped symmetric conformation. But this is a very opposite role, right? FCT actually play a role to trap the receptor in the asymmetric conformation by creating the negative cooperativity, right? So therefore, actually, FCT actually is very important to differentiation the conformation between the IGF-1 and the insulin receptor. So I'll be quick and I'll skip a few slides. So yeah, that is we also saw the structure of insulin receptor with IGF-2 bound, and this because IGF-2 also cannot bound to the site 2 of the insulin receptor, and also there's strong cooperativity between the two insulin receptor site 1 for the IGF-2 binding. The overall structure of insulin receptor with IGF-2 bound is also a similar asymmetric conformation with only one ligand bound as one of the site 1s. Right? So what this means actually, that means when the insulin receptor is stimulated by a different ligand, when it's stimulated by insulin at high saturated concentration, it will adopt this symmetric T-shaped conformation. And when the insulin receptor is stimulated by the IGF-1 or IGF-2, because of the IGF-1, IGF-2 can only bound to site 1 of the insulin receptor, and because of their strong negative cooperativity for the IGF-1 binding to the insulin receptor, so the conformation will be the very asymmetric with only one ligand bound. So you can see that when the insulin receptor responds to different ligand, the conformation actually is very, very different, right? And also we closely look at the conformation of this two, we call it two legs region. The relative conformation between the two legs are also quite different between the two different conformations. And also we actually also, through the lower resolution density, we can also see the crossover point of the TM is also different between the symmetric and asymmetric conformation. So in the symmetric conformation, the TM actually cross over at this N terminal. And however, in this asymmetric conformation, the TM is crossed over at C terminal. Maybe this allows the insulin receptor to respond to different ligand in a different way, and that allows to, maybe this is a way to define the signaling specificity for the insulin receptor. Yeah, but this we still don't know actually. Maybe that is a way how the insulin can tell, oh, this activates by the IGF-1 or this is activated by insulin. Yeah, I have two more slides. So yeah, I told you in the very beginning, so there's a third family of the insulin receptor. This called insulin receptor related receptor. But and so far there's no ligand has been found that can activate this receptor. But there's a study about 10 years ago. Actually, they found the pH can activate this receptor. So if you change the pH from 7 to 9, the receptor can be activated. And people propose actually this receptor play an important role in this allocrine homeostasis in the kidney. So actually, we also determine the structure of the IRR in the apo and the active state by different changing the pH. Right, you can see in the pH 7, the receptor adopts this inverted V-shaped conformation. But if you change the pH from 7 to 9, all the receptor is changing the conformation from the inverted V shape to a T shape. So, but different the insulin receptor and IGF-1 receptor. So those active states are only maintained by the receptor-receptor interaction because no ligand actually are involved in this interaction. Yeah, this is our model. So the pH change can actually disrupt the apo structure and thereby promote the conformational change and for the activation. Okay, yeah, this is my last slide. And so just a brief summary. So yeah, you can see actually, although the three receptor in this family are sequentially a very similar structure is closely related, but the activation mechanism are completely different. For the insulin receptor, it requires the full activation requires full insulin bound to both site 1 and site 2. And for the IGF-1 receptor, only require one one IGF-1 is enough for activation, and the ligand binding to the receptor involve a strong negative activities. And for the IRR, the activation is not, there's no ligand involved, purely by the pH change. Yeah, I already sent most people in the in my talk for my project, and also I want also special thanks to the Korean facility that started is our manager, and thanks my funding agents, and also thanks for your attention. Yeah, thank you.
Any questions?
Yeah, thanks.
Yeah, then, and first I have a question. So actually, among my family and friend who suffering from actually diabetic, and so I was very interested of this insulin signaling. And according to your structure, there is two different sides of binding. And actually, diabetic 2 is aging dependent. With aging, they will develop. And when they are young, they didn't have any issues. With the aging around 60, over 60, they develop diabetes. When you look at your structure, did you see any particular factor with correlate the aging process?
Yeah, I think it's hard to imagine how the agent can affect the insulin binding to receptor. But I think there's a lot of studies suggesting the agent maybe can affect the secretion of the insulin from the beta cells. Yeah, so you don't have enough insulin maybe in the blood.
So you
to activate the receptor. Yeah.
More relate to the concentration of insulin, not actually binding or activating process.
Yeah, that's my speculation. That could be because maybe with the in the senior people, they actually the beta cell maybe it's not that functional compared with young people, right? And it's therefore you may not have enough insulin. I don't know. Yeah, yeah, but this is a good question. Maybe it maybe needs more study. Yeah, I'm not an expert actually in aging. Yeah.
So,
DJ has a question. So I'll DJ, you're on.
So I have a quick question about the downstream effector, right? So you mentioned that for the insulin, it can activate both ERK and PI3 AKT pathway.
Yeah.
But for the IGF receptor, IGFR1, it only activates ERK. So I don't, let me give you some background. Like in Drosophila, for example, in the fly, clearly it can activate both the receptor.
Yeah.
So we tend to think about the fly receptor. We call it IGF like, insulin like receptor. I don't know whether have you done a comparison to look at like some other species and see whether it's more closely related to insulin receptor or IGFR.
Yeah. Thanks. Yeah, this is a good question. Actually, we are working on the fly insulin receptor right now. It's different to fly is different to human. In human, there's 3 different receptors in that insulin receptor in this family. But in the fly, there's only 1 insulin receptor.
Yes.
But also, it's different in the fly. There is 7 different peptides can activate the receptor.
Yes.
And also the signal is also quite different. So my speculation is when the fly insulin receptor is activated by different peptides, maybe they actually have a preference to activate either ERK or AKT pathway. And the conformation of the fly insulin receptor bound with different type peptide maybe is different. Yeah, that's my hypothesis right now. Yes.
That would be very interesting, I think.
Right. Yeah. And in the C. elegans, actually, there's also 1 insulin receptor, but there's more than 30 different peptides can activate the receptor. Yeah. That's actually even more complicated. Yeah, in the C. elegans. Yeah.
Thank you.
Okay. Question?
Yeah,
okay.
Can I have one more question?
Yeah, sure.
And when you look, as far as when I look at the insulin-related, diabetes-related drug development, I don't think still there is a very advanced development. And your beautiful structure and a mechanism could help really precise designing not just a natural insulin, but synthesized insulin. But whether there is a chance that chemically, specifically activate the way developing drug for the diabetics, do you see any approach that way?
Yeah, I think that's a good point. Yeah, I think the SAT1 binding, because it's so high affinity, it's hard to make a better insulin that can bound to SAT1 stronger. But SAT2 is relatively weaker site. So that is still possible to design a better binder that can bound to SAT2 in higher affinity. I think that's possible. But therefore, I think SAT2 is kind of attractive a site for the design a new insulin analog. Another thing, I think also actually we are also working with Dave Baker right now for design those peptides. Some wild idea, because for IGF-1 receptor, there's no SAT2 binding, but in principle, we can design some peptide compound to the SAT2 of the IGF-1 receptor. So in this way, we may convert the IGF-1 receptor to the insulin receptor. Yeah, that is something maybe worth some study in the future. Yeah.
Thank you so much. It was wonderful. And I am very thankful for my family and friends. They will hopefully get help from these studies. Thank you so much.
Thanks so much.
Bye-bye.
Thank you, everybody.
Yeah, thank you.